Hi everybody, my name is Alex Snyder. I am an undergraduate biology major here at Georgia Southern, and I work primarily with social insects. I've been working with my research mentor, Dr. Kevin Loop, for a little over a year now on this project, which we are currently titling Using Microsatellite Loci to Determine Colonial Social Structures to Better Understand the Multi-Queen Behavior of the Southern Yellowjacket. Social insects have always been one of the central focuses of behavioral biology research throughout the existence of the field. Novel behavioral traits, like the social structure of the southern yellowjacket, have always warranted much study. Southern yellowjacket, or Vespula squamosa, is known to utilize two different social strategies. Most wasps, the southern yellowjacket included, will establish a single queen nest in which a single queen will construct a nest in the spring that usually lasts until late fall before the workers eventually die out, and the daughter queens will go to overwinter or hibernate until the next spring. This annual strategy is by far the most common among most social insects. However, Vespula squamosa also uses a multi-queen colony strategy as well. These colonies typically follow a similar first year as the single colony or the single queen relatives. The difference being is that the multi-queen variants, the worker population is usually reduced during the winter months, but is not killed off completely, as new queens, either from the same or different colonies, begin to reproduce in the nest. These multi-queen colonies in their second year can grow to enormous sizes and contain thousands of workers and hundreds of queens. Some multi-queen nests have been found in Georgia and Florida. However, little is known about their presence in the southernmost parts of their range, like Mexico and Guatemala. The end goal of our project is to better determine the true extent of how often multi-queen colonies occur in these ranges through the use of DNA analysis using segments of DNA called microsatellite loci. So 8 wasp nests were collected from Santiago Apuala, a town just northwest of Oaxaca, Mexico. They were collected by Nathan Durstein, Sebastian Jimenez, Gerard and Regina Grise. DNA extraction was performed on an average of 36 individuals from each of the 8 nests. After performing DNA extraction, polymerase chain reaction, or PCR, was performed in order to amplify the DNA of the selected wasps at 8 highly variable microsatellite loci. Depicted below is a output chart from one of the program's gene marker, which we use to sort and interpret the outputs of the genetic analysis project. The intention is to use the data collected to form what is essentially a family tree of the queens and workers in the nest through a statistical analysis program called Colony. The collected data was then used to determine the most likely number of queens in each nest. From this data, we found that there were about an average of 5.5 queens per nest. The nest with the lowest number had 3, and the nest with the highest had 11. So, out of all 8 of the nests, we noticed that multiple queens were in every single one of them. Even though our sample size was fairly small, it is likely that the multi-queen social strategy is actually fairly common in this area of the Vespula squamosa home range. This is interesting because Vespula squamosa is the only known yellowjacket to create multi-queen colonies within their native range. Other yellowjackets only make multi-queen colonies outside of their native range, usually as invasive species. This suggests that the multi-queen social structure in other wasps may not be as a result of the fluke of bottlenecks or environmental mismatches that usually come with the invasion process. We can tell this because we have observed evolutionary stability in the multi-queen colonies in the Vespula squamosa, as these nests have existed at a relatively stable rate for a long period of time. These multi-queen colonies may even exist in other yellowjackets within their native range, and we just haven't found them yet. In the end, I think a lot more work will need to be done in order to better understand both how these multi-queen colonies have evolved originally and to the extent in which these multi-queen colonies exist in the Central American portion of Vespula squamosa's range. Thank you for watching my presentation. I hope you enjoyed it.